I'm done.
You do? You should try it.
That hurt.
But it wasn't. It wasn't.
That hurt. It didn't hit like muscle did it. Right on. Full speed.
Did everybody get your unknown onto sporulating auger and Indian incubator? Wanted to quickly show you a couple of slides because I've had people come in and say, I'm really not sure what I'm looking for on the negative stain. And this is a slide somebody did last year of their unknown for negative stain. And again, what you can see on the negative stain is that nigrosin, which is an acidic stain, will attach to the slide and not the bacteria, because the bacteria and the stain have the same charge, so they repel one another. So what you're looking for here is these clear areas represent the cells. Okay, so these are some bacillus bacteria, and here you can see the division between those two particular cells. So each one of these would be an individual cell. They don't all look that nice and pretty. Here's another negative stain slide. Still bacillus, you can see pretty well. But then here's some cocci right here. And so if you have one of the smaller, either cocci or the short little toxobacilli, it can be a challenge, okay? Do the best you can, okay? Play around with the light on your microscope, play around with the iris opening, and just get the best possible image you can. I was just telling Kendall, sometimes they actually look a little bit better once you throw them into a PowerPoint than if you're just looking at them on the screen of your phone or the iPad. And by the way, something I didn't have a chance to tell you yesterday is that one of the fatal design flaws of this camera right here is that it doesn't have an on-off switch, and I don't like to leave it on all the time. So the way you turn it on and off is you just unplug the power from this little block right here in the cord. So if you come in and it's not doing anything, the power's been unplugged, okay? So just be aware of that. Anybody know what lab we're doing tomorrow? Okay, we're doing the stain, the acid-fast stain tomorrow. You remember exam coming up, what acid-fast stain is differentiating and looking for? Cell wall. There's a substance called mycolic acid, which is a fatty substance at the cell wall, particularly of mycobacterium. And during the acid-fast staining process, those particular cells stain a particular color versus the non-acid-fast stain. Again, this is a little bit hard to see right here, but there's actually two organisms mixed together here on purpose. There are some rods, which are more pinkish, reddish color, and then there are some cocci, which are blue. During the acid-fast staining procedure, the acid-fast positive bacteria retain this primary red dye and therefore give that reddish purple color, and the acid-fast negative bacteria don't retain that dye, and then you counterstain them with methylene blue, which gives them a blue appearance. So red versus blue is your kind of outcome for acid-fast staining. Okay? This one, we've got to do some steaming, which will involve driving the stain into the cell wall. And so make sure you look at your instructions and see what you have to do. But we're only going to set up one slide, and basically we're going to have three organisms on the same slide the way we did it for the gram stain. So you'll have your unknown, we'll have a mycobacterium, which is a positive control, and we'll have a staph, which is a negative control. So you'll be working with a partner to do the staining procedure, but each one of you will be doing your own slide. Okay? Any questions about tomorrow? Okay, a little review. We talked last time about the standard plate count as a way to determine numbers of microorganisms in a population. So if I've got a little 100 ml flask here of bacterial solution, and I want to know the concentration in cells per mil, So what we did is a series of serial dilutions, and so this tells you how much was taken out of the original culture, and then what it was put into in terms of a dilution tube. Then this amount was taken and put into this amount, this amount, this amount, this amount. Okay? I've done one of the calculations for you. I've calculated the step dilution at each one of these individuals, and then the total dilution. Okay? And then from each of these tubes, we took 1 ml of this solution and put it on a plate, incubated it, and then this is the number of colonies that we got. Okay? So given this information, can you tell me concentration? Shot. Write down your answer. You think you come up with it. Anybody think you got the answer? Anybody come up with that answer? Okay. What are the key elements you need to know? Let's just take a look real quickly here. Why is this a 10 to the minus 1 step dilution? Because we took 1 ml and put it into 9 mils of fluid. Remember, the formula is number of mils of dilute divided by dilute plus dilute1. So 1 over 1 plus 9, 1 over 10, 10 to the minus 1. This one, 0.1 into 9.9, that's 1 to 100 dilution. And so that's a 10 to the minus 2. Your step is the product of those two. Okay, so this was the first dilution, so that was 10 to the minus 1. But now the step here is the product of those two. Okay, another 10 to the minus 1 here, so your step is 10 to the minus 4th. Another 10 to the minus 1, your step is 10 to the minus 5th. Here, 0.01 into 9.99 is 10 to the minus 3, 1 to 1,000. Okay, so that would be 10 to the minus 8th. So you're going to have to do this again later on in lab, but knowing how to do those dilution factors is important. You will have to know that for the lab exam. Okay, what's the only countable plate in the series? Which dilution? Yeah, the 50 colonies, the 10 to the minus 4th dilution. Remember, 30 to 300 is considered to be countable. So you do have colonies here, but this is too few to be statistically significant. And obviously nothing here. Too many here. Okay? Here's your formula. CF, which is basically the number of colonies, is equal to the initial concentration, which is what we're trying to solve for, and DF is the dilution factor. So if we want to solve for this, basically we just divide colonies by dilution factor, 50 by 1 times 10 to the minus 4th, and that's the same thing as 50 times 10 to the 4th, or 5 by 1 times 10 to the 5th. Okay? What would be the advantages and disadvantages of using this method to count microorganisms? What's the one big advantage this has?
Cheap.
Okay, cheap. There are other cheap methods that don't have this advantage. Yeah, this gives you a viable cell count, okay? Because the only things that will give you colonies were the cells that were alive. What are the disadvantages? Potential for contamination, what were you just asking? Diluting is okay if you do it, properly. So it's prone to error, okay? And you're making an assumption that each one of these dilutions represent, in this case, a 1 to 100 dilution of this. If your pipetting's off, if you don't get things mixed up properly, then those dilutions aren't really valid. These numbers here make sense in terms of dilution factors, but you wouldn't get nice clean numbers like this, okay? They would be much more variable than that. So this has its applications, but it also has its disadvantages. Okay, we were talking at the end of the period about using turbidity as a way to measure cell populations. This one right here?
Yeah.
That is this total dilution at this particular point, which is the product of each of these step dilutions. Okay? So I said last time, okay, we've got these tubes right over here. The one on the right obviously has more bacteria in it than the one on the left. It is more turbid. We can actually tell relatively how many times greater the cell number is in this one than this one by using a spectrophotometer and taking absorbance readings. But we cannot get a concentration even from just spectrophotometer readings. Okay? So how can we do concentrations, which gives us actually cell numbers from data based upon turbidity? Well, to do that, you have to construct a standard curve. So what do we want to know here? We want to know the relationship between absorbance and cell numbers. It would. Yep. So that's one of the things that we'll talk about in just a second is standard curves are only applicable for, you need a different standard curve for every bacteria. You need one for a particular type of growth media, so it might be different in broth versus something else. You need one based upon a particular temperature, so that'd be growing at 37 degrees, for example. So a standard curve might be generated for E. coli growing on tryptic soy broth at 37 degrees centigrade. Okay? When we're making our standard curve, what goes on the y-axis, what goes on the x-axis? What's the dependent variable? That goes on the y, right? Which depends on which? Does absorbance depend upon cell number or concentration, or does concentration depend upon absorbance? Absorbance depends upon the numbers of cells, right? The more cells you have, the more absorbance there is. So here, absorbance, concentration. So we make a standard curve, and the standard curve will typically level off because at some point your absorbance values just are too great. You can't see any change. You can't really measure them. So how are we going to make the standard curve? How are we going to know the cell numbers if I take a particular culture and pop it into a spectrophotometer and it generates a certain absorbance reading? Yeah, I'm going to have to use one of these other methods to count the microorganisms. I might have to do a direct microscopic count, or I could do a standard plate count. But once you have that standard curve generated for a particular organism under particular conditions, then if you grow those organisms under those conditions, and you know what organism it is, and then there are published standard curves available for each one of those, then you can just take absorbance readings and then translate that into a particular cell concentration. Okay, so somebody's got to do the work first to do a standard curve. Okay. But once that standard curve is done, then you can just utilize that particular information given this particular caveat, okay? And not all standard curves apply to all microorganisms. Well, this whole concept of a standard curve gives us other opportunities to measure cell growth using other parameters besides turbidity. When microorganisms grow and metabolize, you're going to see a relationship between metabolic rate of the culture and number of cells there. Number of cells, more metabolic rate. And so other strategies you can use for a standard curve, instead of plotting absorbance versus concentration, is if the culture is depleting some substrate, for example. If you can measure the depletion of that substrate, then that could be your independent variable. Or product formation. Are they producing some product? What would be a good substrate depletion variable that you might use? What's something that's used during metabolism? Oxygen. Okay, so you could look at oxygen depletion rates of the culture. And we could use one of the little vernier probes, one of the little oxygen sensors, and put it in a culture over time. But again, somebody would have to do the work of saying this particular oxygen concentration at this time under these conditions would be the result of this many cells. Okay, so you'd have to use one of those other counting methods to generate your standard curve. What would be a fairly easy product formation to measure? Again, using our vernier probes. Oxygen is depleted during aerobic respiration. What's produced? Carbon dioxide production, okay? Or in fermentation, it might be ethanol production, okay? So there's several different parameters you could use and measure to generate a standard curve. Again, the advantage of this is that it's a lot of, the disadvantage is it's a lot of work to set up the standard curve. But once the standard curve is established, it's a relatively fast method to determine cell concentrations. One more example, and this will be the breaking point. Let's use a medical application this time. Sometimes it may be of interest to not know just how many cells there are in a particular population, but how many pathogens there are. Okay, we don't worry about the non-pathogenic bacteria. We just want a way to measure the number of pathogenic bacteria in the sample. And a strategy you can use to get some information about pathogens is try to measure disease production in test animals. And probably likely in most of these tests you wouldn't actually be using animals like rats or mice. They get pretty expensive. You might be using, protozoans, for example, which can be cultured easily, really cheap. But the key here is that those particular pathogens have to be able to produce some recognizable symptoms in your test subject. And one of the symptoms could be death. But it might be something else. It might be a lesion of some type, but some symptom that is specific to that particular pathogen. There's a technique called the 50% endpoint analysis that does this particular type of measurement. And in this particular technique, again, you have to do a series of serial dilutions of your solution. So let's say I do a 10 to the minus 1 dilution, a 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. Okay, so this is a dilution of your original solution that you're measuring. And then from each of these dilutions, we inoculate, let's say, 10 test subjects. And then we look for the presence of symptoms associated with that particular pathogen we're trying to measure. And we might get results that look something like this. Does this inherently make sense that the number of subjects showing symptoms would decline with a greater dilution? Okay. The more you dilute something, the less pathogens there would then be present to cause diseases. Well, this allows us to calculate the 50% endpoint dilution. And what is that? That's the greatest dilution that gives symptoms in at least 50% of the test subjects. So what's our 50% endpoint dilution in this example? 10 to the minus 3, okay? We're getting more than 50% here, but that is not the greatest dilution that gives us 50%. So here our 50% endpoint dilution would be 10 to the minus 3. Now, this doesn't really give us a concentration. This gives us something called a titer. And we'll talk about titer later on in the context of immunology. But titer is kind of a relative measurement. It's a comparative measurement. And the titer is the minus log of the 50% endpoint dilution. So it's minus log of 10 to the minus 3. You know, what's the minus log of 10 to the minus 3? Great. So do things that have higher titer numbers or lower titer numbers relative to one another have more pathogens? The higher the titer number. Because if I've got something with a titer of 5 versus a titer of 3, what does that mean? That means here I can dilute it 10 to the minus 5 times and still get my 50%. Here I can only dilute it 10 to the minus 3 times and get my 50%. So this had less cells in it to begin with than this did. Make sense? So the higher the numerical value of titer, the more pathogens that could be present. Because this means the solution can still be diluted more than this one did to still give you that same 50% endpoint result. So there's a number of different ways to measure microorganisms, and it kind of depends upon what your situation is and what your resources are. The real advantage of this one is that it allows you to measure specifically pathogenic microorganisms. What are the downsides of this particular method? Pardon me? Yeah, you really can't tell necessarily which one is which. You might have more than one thing. Now, if you're working from an isolated colony, that would be okay. All your problems with making dilutions, same thing you had the standard plate count. But what's the biggest drawback of this, really? Just think about the logistics of this, okay? Having to have these groups of 10 test subjects, okay? And if you were using animals, that would be expensive. And then it's going to take time for the symptoms to develop or the organism to die. Okay, so I'll put a big line in your notes. And this represents the break point for the exam. I think of the 2 exams that I sent you, the last year's exam, I think actually went right up to this point. I think the other one that I sent you, I think stopped before we got to the counting section. So it just kind of depends on rhythms of the semester and how far we got. But this will go through this particular information. And then on the study guide, I think it's about halfway through the study guide. The next topic we're going to talk about is factors that influence microbial growth. So on that study guide for growth, if you see questions about that, that's next exam. Okay? As far as exam is concerned, I mean, you'll look at the old exams, you'll see what they were like in the past. They're much like the cell biology exams. There's some multiple choice questions, there's some essay questions, there's some agree and disagree questions. And we really need to be time sensitive. And so you can come in here and start at 10 till, and you've got to finish by the top of the hour. Okay? So plan your time accordingly. Any questions about the exam? Okay. So now you've got some insight into how we can go about measuring microbial growth and counting the number of microorganisms. Let's spend a little bit of time talking about some of the factors that influence microbial growth. And we've mentioned some of those already. Specifically, we mentioned temperature, because one of the things that you did was you determined a temperature optimum for your unknown. We made some reference to pH. For photosynthetic organisms, availability of sunlight is going to be a determining factor that influences rate of growth. We're going to spend a fair amount of time talking about gases that influence rate of growth, and particularly oxygen and toxic byproducts associated with the metabolism of oxygen. And that's going to be an important point that we'll spend some time talking about. We also will talk a little bit about osmotic concentration. And these are all things that we're going to follow up doing in lab. You've already done the temperature optimum. One of the things that we'll do in a couple of weeks is you will determine kind of a general solute concentration optimum for your unknown. And that will be diagnostic information. That will be important. Okay? But we're going to spend a fair amount of time talking about oxygen and determining your organism's oxygen requirements, because that's something that's really going to be useful in keying out in Berkey Spaniel. Is your organism aerobic? Is it facultative anaerobic? Is it microaerobic? Is it aerotolerant? And what does that all mean? What are the implications for the organisms of having these different sensitivities to oxygen? This table is just kind of interesting. This gives you a general analysis of the chemical composition of an E. coli cell based upon total weight and dry weight. Obviously, total weight, there is water that makes up a big percentage of the cell, about 70% of the cell. Dry weight, then, water's not accounted for. So you can see lots of protein, RNA, a little bit of DNA, some carbohydrates, everything else kind of mixed together. And then this shows you elemental composition. And carbon obviously is pretty important, organic molecules. A lot of oxygen, nitrogen's fairly important, hydrogen, and then there are these various elements: phosphorus, sulfur, potassium, sodium, chlorine, iron. This one's kind of hard to see. This talks about where microorganisms can get some of their key nutrients. Whoops. The carbon, the hydrogen, the oxygen, phosphorus, etc. And one of the things you're going to appreciate about bacteria is bacteria are very adaptable as a group about how they can get their nutrients. For us, all of these main nutrients come primarily from organic compounds. We get our carbon from organic sources. We do, as individuals. We get our nitrogen from organic sources. Oxygen, we obviously breathe from the atmosphere. Hydrogen, we can get primarily from organic compounds as well, fats, for example. And then these elemental things, phosphorus, sulfur, potassium, we have to get from other sources. They might be from vitamins, they might be from plant material. But with microorganisms, there are all these other possibilities where some of these things can be obtained. And that's something that we're going to delve into more when we talk about microbial metabolism. We are using a fair amount of culture media in this course. The major media that we use are TSB, which stands for tryptic soy broth, and TSA, which is tryptic soy agar, nutrient broth, and nutrient agar. These are the two we made during the first lab. These are typically what we use routinely to culture our organisms. We buy bottles of this stuff that are dehydrated. Does anybody remember what was in the nutrient broth that we mixed together? You're over here at the balance weighing things out. What were you weighing out?
Beef extract.
We were weighing out beef extract. And for the last couple of years, we've been using a powdered beef extract. I used to have this old nasty container of this kind of pasty material. You need to kind of scoop it out of there, and it was all messy. Beef extract is produced literally by taking meat and just boiling it down and decomposing it until it becomes this kind of pasty stuff. Now they dehydrate it, so it's a powder. Beef extract is primarily your source of various vitamins and minerals that bacteria need to grow. So on this particular list here, it might contain some of the sulfur and phosphorus or zinc. Okay? What else is in that mix besides beef extract? What else did you mix together? Sodium chloride? What was the sodium chloride intended to do? I suppose you could say, well, it might be a source of sodium and chlorine. It's got another major purpose. The amount of sodium chloride that you put in there was intended to make that particular solution when you rehydrated it isotopic. So that's its primary function. What else did you put in nutrient broth? There was this powdered stuff called peptone. And basically peptone is the source of carbon and nitrogen. It is digested protein. So basically it's a mixture of amino acids which contain both nitrogen and carbon. The real difference between nutrient broth and tryptic soy broth is what's the source of protein that's digested to give the carbon and nitrogen. Here it's soy protein. Okay? In peptone it's, I forget what it is, what kind of protein it is. That's why they call this tryptic soy broth. What's the difference between nutrient broth and nutrient agar? Nutrient agar has the solidifying agent agar in it. Okay? Well, it's really pretty remarkable that bacteria can grow basically, if you give them those four things and water, and then put them at the right temperature, at the right pH. And as I said earlier on, I could stick you in a closet and feed you agar for the rest of your life, and you wouldn't survive. We don't have the metabolic capability to make everything we need to make to be a cell, all of this stuff right here, out of those basic ingredients. We have to get some of these kind of pre-formed in our diet, particularly some of those vitamins and cofactors. We just can't make. We don't have the metabolic machinery to do that. The bacteria do. Now, there are some bacteria that aren't so good at growing on this material that we call minimal media. So these are all considered to be minimal media: tryptic soy broth, tryptic soy agar. Okay? Organisms that can't grow on that minimal media are said to be fastidious. And they require some supplements. There's some things that they just can't make. It might be a coenzyme. It might be a particular amino acid that they need. They just can't synthesize that particular amino acid. Can we synthesize all 20 of the amino acids? No, there's some that we have to obtain in our diet. What do you call those? Those are essential amino acids. Okay. But other amino acids, once we have one amino acid, we can sometimes convert it into another one. There is one fastidious organism called Haemophilus, which causes a common type of influenza, that requires heme. These organisms won't grow on minimal agar plates. We have to supplement them with blood. And we're going to make some plates of blood agar later on. And Haemophilus will need to be grown on blood agar because they need heme. They can't manufacture heme, and if you can't make heme, you can't make the cytochromes that are part of the electron transport system. They need the heme, they need the whole group. Now there are some bacteria that just require additional iron, and they typically grow on blood agar too because they actually produce an enzyme which lyses the cells and breaks down the hemoglobin and then liberates the iron, and they use iron. Well, they need, it's a micronutrient, and it's needed in some of the electron transport chain compounds. There are other compounds that require iron. Iron oftentimes is a limiting factor for microbial growth. And one of the defense mechanisms that your body sometimes employs when you get sick is that there are some mechanisms that kind of scavenge up iron out of the blood. And then if there's not enough iron available in the blood, bacteria won't grow. If they're particularly sensitive to iron levels, that's a way for your body to kind of kill pathogens. Here's a snowfield, and you can see some reddish stuff here, and that's not a bleeding hiker. That is actually a microorganism which is photosynthetic, which can grow under cold conditions. Those particular organisms belong to a group of organisms that are called psychrophills. So when we look at temperature profiles of organisms and what temperatures they can tolerate and which ones they thrive at, there are three general patterns of microbial temperature optimum. There are the psychrophills, the mesophils, and the thermophils. What does meso mean? Middle. Okay. Mesophyll is the tissue in the middle of a leaf, between the two epidermis of a leaf. So mesophils have this temperature optimum kind of in the middle. When you talk about temperature considerations and influence of growth, there's really three values you need to be aware of. What is the minimum temperature at which something can grow? What is the maximum temperature at what it can grow? And then what is the optimum at which it can grow? So mesophils run this range from 10 degrees Celsius up to about 45 degrees Celsius, where you'll see that psychrophils have a much lower temperature minimum, maximum, and optimum as well.
It looks like the roller goes to the right, almost like that. Whatever drop off after they
Yeah, and I think that really has to do with, Zach, primarily the effective temperature on denaturing proteins. So you get a growth bump because you get increased metabolic rate with increased temperature. So speed of chemical reactions by heating them. But once you get past that optimum, you get a fairly fast drop off because of denaturation of proteins. That's a good observation. And then the thermophils have a much higher temperature optimum. Okay? So what category do all of your unknowns fall into? They're all mesophils, okay? Because we're incubating at 25 to 37 degrees. And 37 degrees Celsius will be the optimum for many of you, particularly if your organism is an enteric organism. Remember what enteric refers to? Family Enterobacteriaceae, the enterics. Sometimes called coliforms as well. Those are your gut bacteria. Okay? They're things that live especially in your large intestine. What's your body temperature, ideally? Celsius. 37. Okay? That's around 98.6. So any of the enteric organisms probably would have that 37 degrees Celsius temperature optimum. Sometimes the temperature optimum of organisms is pretty wide. Staph has a very wide temperature optimum, or temperature range, sorry, not optimum. And the temperature range of staph really can encompass this whole range right here. Some organisms have a very narrow temperature optimum. I mentioned Mycobacterium leprae. It has a temperature optimum between 33 and 35 degrees Celsius, where staph is about 10 to 40. Okay? Mycobacterium, because of its restricted temperature range, really is only able to grow on the extremities of tissue, for example, of the host that infects. So the face is affected, hands, feet are affected, but typically the core body would not be affected by a Mycobacterium leprosy infection because that would be above the temperature range that can tolerate. There are a group of organisms that are said to be facultative psychrophils. And these are organisms that are actually technically mesophils, because their optimum is one of these middle type of temperatures. This would probably be right down here, this little green area right here would be a facultative psychrophil, because its optimum is a much higher temperature, but for periods of time they can tolerate colder temperatures. So it's almost like they're kind of in this gray area between psychrophils and mesophils. Why do you refrigerate things? You don't want them to spoil. But how does refrigeration retard spoilage? Slows it down, slows down metabolism. Okay. And at some point, if you get the temperature too cold, then water starts to freeze, which creates a problem. And what's particularly a problem is if that water thaws then, That freezing and thawing can create ice crystals, which can tear the cell apart. But facultative cycrophills can kind of get by in the refrigerator, and if you leave them there long enough, they will grow at a very slow rate. So when you pull that nasty thing out of the back of the refrigerator that's been there forever, and it's covered with kind of a slimy substance right there, that was probably a facultative cycrophyll organism. There is another type of organism that is called thermoduric. And this is also a mesophyll. But thermoduric organisms can tolerate brief exposure to higher temperatures. There are some strains of E. coli which are thermoduric. And this can be a problem if these E. coli are the strain that is hemorrhagic. Those can be lethal. Thermoduric organisms are the reason why you've got to be careful when you cook beef or other meat, particularly ground beef, because when they make ground beef, they kind of grind everything all up, and bacteria that were in the cow's intestines can get mixed up with all the other meat. Well, normally when you cook, if you cook to the proper internal temperature, then you'll kill even those thermoduric organisms. If you like your meat really red and bloody and rare and things like that, that temperature might not be sufficient to kill those thermoduric organisms, and they could survive and cause problems. So there's some kind of organisms that are kind of in the middle, kind of the twilight zone between mesophils and thermophils, or mesophils and cycrophils. Well, you can get similar temperature similar profiles if you look at pH optimum of cells. And again, pH optimum, organisms will typically have a range of minimum pH they can tolerate, maximum pH they can tolerate, but there's typically an optimum pH as well. For most bacteria, the pH optimum is in that kind of 6 to 7 range. Fungi prefer a little bit more of an acidic environment. Oftentimes the pH optimum of fungi is in that kind of 5 to 6 range. If you were using plates of growth media, because your TSA plates are buffered to be around pH 7. If we used a fungal growth media, which was around pH 5, there's this stuff called Sabarus dextrosager. That really supports the growth of the fungi much more readily. Think about the contamination we got and what we saw growing on some of those plates, even on an agar plate at pH 7. That does not represent all the fungi that were present in the atmosphere. Most of them didn't grow. If you use that Sabarus dextrosager instead, you would see far more, diversity of fungal pathogens. You wouldn't see as many bacteria though, because then the pH optimum would be a little bit low for most bacteria. There are bacteria that can tolerate pHs as low as 2. There is a bacteria called Heliobacteria pylori. Anybody know what that's causative agent of? Stomach ulcers. Okay? And they have adaptations that allow them to exist in the highly acidic pH of the stomach. Why do you have a stomach? Okay, there's a little bit of digestion that takes place. Okay, there is some pepsin in there, which will break down proteins a little bit. There's a little acid digestion, but basically the reason for the stomach is twofold. It's a way to store food so you don't have to eat constantly, but it's a vat of acid which kills most of the bacteria that you consume when you consume food. Okay? So how can bacteria survive in that acidic environment? Well, there are all kinds of adaptations. We can talk a little bit about some of those when we pick up Tuesday. So read your lab. Acid fast stay is pretty short. You're ready to go.
Alright, I'll get out of lab.